Good morning, everybody. So we'll continue the next topic, which is so we looked yesterday about the polyspecificity, and now we can look at the mechanisms. And so we also covered the glucose transporter as first day as types of transporters. And so this is one of the most studied transporter, which also is facilitative or passive transporter and belongs to SLC2 family. The recent report says there are 1 to 14 transporters, but not all of them have been characterized yet. Okay, and if you look at the red blood cell glucose transporter, this is one of the major component of the red blood cell membrane, which is like 5% of the membrane protein is the glucose transporter, because as you know, the red blood cells do not have mitochondria, and so they cannot generate ATP by using respiratory chain. So they have to depend on bringing in glucose and glycolytic pathway. The same way, most of the cancer cells, if you look at their energy source is really glucose and glycolytic pathway. So you must have heard about a term Warburg effect, which is really again similar where you have the higher metabolism of glucose, and the side effect of that is you get acid secretion, and that's why in tumor environment you'll see there is more acid secretion compared to normal tissue, and that also is the reason as you go from periphery of the tumor to the center of the tumor, you have hypoxia, which means there's less and less oxygen, lack of oxygen, and you keep having this increased acidity because you release lactic acid from glucose metabolism, and that's Warburg effect in nutshell. Okay, so this is typical transporter topology, and the specificities are given here. The blood tissue barrier. So the GLUT1 is mainly the housekeeping gene where you need to have low levels in many tissues glucose. You have the GLUT1. GLUT2 is present in liver, pancreatic, beta cells, absorptive tissues, and in liver it is both present on the canalicular membrane as well as basolateral, because you need to have bidirectional glucose transport. Then GLUT3 is in brain, nerve cells. GLUT4 is in fat, muscle, heart. GLUT5 spermatozoa and small intestine. And the specificities, some of them are highly specific to the glucose, D-glucose compared to the others. And GLUT2 actually also would transport like galactose and other sugars. And this I think we already covered on Monday, where you have the glucose, high concentration of glucose in the plasma or medium, and then your binding sites are facing outside, and this is essentially the same alternate access mechanism where you have the bound glucose, which then has the conformational change and you release the glucose inside. And this is just equilibration because there is no concentration of glucose against the gradient. So moving on to the sodium glucose transporters. So you will see the difference between the structure. Here you can see this is 12 transmembrane helices, but there are these are hydrophobic amino acids as well as some polar residues within the transmembrane helices. But when you come to the sodium glucose transporter, one interesting observation is that in this family you will see that transmembranes like a 2, 3, 4, 5, 6, these which is shown in the cyan box and the pink box, they have exactly inverted topology. So this is, if this is the orientation, in this case you have the reversed, and this has been found in a number of transporters, and that seemed to be the reason seem to facilitate alternate access mechanism. We'll also come to another mechanism which is used, which is elevator type mechanism. I'll also show you that. This is the crystal structure of the bacterial sodium glucose transporter. This SLC5 family has 12 members, and glucose galactose are substrates for the sodium glucose transporter 1. Yes. So we'll come to here. So essentially what it means is that when you have a gradient or a higher concentration on the one side, so in this case, as we discussed earlier, there is a membrane potential which is positive outside and negative inside. And then first the sodium binds and then the glucose binds, because this is a sodium coupled sugar transporter, unless sodium binds, the glucose cannot bind because there is a conformational change. And once both of them are bound, as you can see now, this binding site is now flipped. So that's the alternate access mechanism, from out to in or in to out. Okay? Because as you can see, when you have one site being occupied here in the same orientation, you don't have another site accessible here, right? So at a given time in a single transporter, only one side the sites are available. And in this case, because this is a co-transporter, you have both two molecules binding. One is sodium, one is glucose. But the same thing is true even for a single glucose transporter where you transport glucose by diffusion. It's because that site, glucose site, like when we saw it in the previous one, see, it's not available at the same time on this side. What you have the site available here. And that's why it's called alternate access. Okay? So you have the sodium and glucose bound, you release glucose first, and then you release the sodium, and then you can go back and forth. So this would be the cycle for sodium glucose transporter. It also works for other sodium coupled systems like sugar, amino acids, and others. Essentially, it's the same. In this case, as you can see, there is a total one charge is moved, one positive charge, because the glucose is electroneutral, doesn't have a total overall charge.
Sir,
yes.
We know that once glucose enters in the cell, it gets immediately phosphorylated.
Yes.
With hexokinase.
Exactly.
So if some defect happens in hexokinase translation of it, so is there any possibility that it is going out of the cell, through the same channel?
It will depend on the gradient of the glucose.
Okay, the concentration.
Yes. Yes. And also, it is not clear whether under this condition the intracellular glucose will be able to bind, because as you said, there is no real free glucose available in cytoplasm, because as soon as the glucose is released, it gets converted to glucose 6-phosphate. But if you inhibit the glucose 6-phosphate, the hexokinase, I'm sure you will accumulate. So that's another way. On Monday I was telling you that one of the analog of glucose is used, is 2-deoxyglucose. But that 2-deoxyglucose actually gets phosphorylated to 2-glucose 6-phosphate by hexokinase. But other like L-glucose will not get phosphorylated, and that will accumulate. But it's in liposomal system, you can show that then the glucose on this side bound can be transported, but not in the in vivo situation, because the gradients don't work that way. Otherwise, it will be a futile energy cycle, right? If you bring in glucose and again release it outside, there's not going to be anything useful. And those conditions are called futile cycles, where you just use the energy to exchange the ions. Those people do mostly under experimental conditions, where you add C14 or tritiated glucose, and inside, let's say outside you add micromolar tritiated glucose. Tritium is radioactive component, and you'll have say 10 millimolar unlabeled glucose inside, and then you can show the exchange of tritiated glucose coming in, and that is just a way to look at the function of transporter as an exchanger. But there is no net movement of glucose, okay? And that's why it's called futile cycle. So on the other we also covered these peptide transporters on Monday, but these are specifically for the oligo-di and tripeptides. It's mostly present in intestine where you have the absorption of proton digestion products. Protein driving force here is proton gradient rather than sodium gradient. And this is more involved in your beta-lactam antibiotics, ACE inhibitors, renin inhibitors, thrombin inhibitors. But most of these actually you will see only cartoons because people have not yet studied what are the binding sites for peptides or what residues are involved. So we don't really know much about. So if you look at whole transporter field, you can actually count the number of transporters for which the mechanism is known on your fingers, because those are model systems. But since they belong to similar families, you're not going to have that many different mechanisms. So it's redundant studying in every transporter unless you see something different. Okay, so now this is something different. So I told you earlier, the secondary transporters you have alternate access, but then interestingly there is a family of transporters in bacteria which actually transport, like say succinate or other TCA cycle intermediates or amino acids. And here what happens is, it's interesting that each transporter monomer has a rigid domain, this blue, which is called oligomerization domain. And then you have a mobile transport domain, this orange. The sodium binds these three pink dots you can see, and then the succinate would bind. But now instead of having that alternate axis where the transport site is moving in, what happens is it just slides against this rigid domain. Okay, you can see the difference now. It will slide and then now release into the inward facing. So that's why it's called elevator, or here you would call lift mechanism, because all you are doing is bringing it down one floor and then going. So this is a very new recent study which is just in last year or two people have found. So you find these interesting oddball mechanisms. We don't know yet examples of this transporter in mammalian system, but it's possible that there are possibility that there might be some. So this is sort of a unique example. Okay, and then the other among the secondary transporters, we saw on Monday that only exception, instead of the influx or uptake transporters, this family where you have the SLC47 multi-drug and toxin extrusion system is actually functions as an efflux transporter instead of bringing in the things, because as you can see the substrates are toxins and multi-drugs. It doesn't make sense to bring them in rather than you want to pump them out. And there are two examples, like at least two members. MATE2K is one of the isoform of MATE2. Mostly they are present in kidney, liver, testis, colon, kidney. And co-transport mechanism, just as we saw with the sodium glucose transporter, driving force is the proton gradient, and then some of the substrates are like the metformin drugs, the TAE, and inhibitors, quinidine, cimetidine, and all. But some of the recent reports show that because there is a nephrotoxicity of cisplatin, they think that maybe the cisplatin is affecting these MATE transporters on the apical surface of kidney, and that's how it gives you nephrotoxicity. And so we will be discussing this in the second session, that just last month the FDA came out with the new guidelines for transporters, and they have now included MATE1 and MATE2K to the list of transporters which should be included in your study when you study the drug-drug interactions. So this is the latest, just less than a month ago. Okay. Look at now the ABC transporters, but mostly we are focusing on the transporters which actually are relevant to this course, which is those which transport anti-cancer drugs or amphipathic compounds, and even the natural products which are amphipathic compounds. So, so far we were discussing, except for the mate family, which has some of the toxins and multi-drug which are amphipathic, mostly water-soluble compounds like glucose, amino acids, and nutrients and others, or even ions, right? So so the difference you can see here is when you have a pump like sodium potassium ATPase or calcium pump, the membrane really, you don't see any of the ions or the solutes being partitioned into the membrane. So either you have them outside the membrane or inside the membrane, and then there is a channel or the alternate access mechanism by which they are either brought in or pumped out. So in this, the membrane doesn't play any role as such, because these are charged compounds. But the minute you come to the amphipathic or hydrophobic compounds, you can see now you have 3 compartment system. You have your substrates outside. Now they partition into the membrane. Even most of them actually partition more into the membrane than in the intracellular region. And then, so there are evidences showing that most of these get pumped out from the membrane itself. And we discussed yesterday polyspecificity, broad substrate specificity. So to put this together, people have proposed this hydrophobic vacuum cleaner. Just like your dust, the motorized vacuum cleaner, all the transporter is doing is whatever is nearby in the membrane, it gets pumped out. And that's why it's called vacuum cleaner. Because when you clean with vacuum, you are not really differentiating what particles or whatever is coming, except for size maybe to some extent. And that's the real way to describe this transporter, especially the P glycoprotein. But to study this kinetically is very difficult, because you don't know what the concentration of your drug is in the membrane. You don't know how much it is in the outside and how much it is inside. And so when we describe affinities, we always say apparent affinities, otherwise apparent Km, apparent Ki, and those, because we have no control over this. Then the another version of this model is where many phospholipids, when they get synthesized, they get transported to different membranes by this you must have heard the flopping and flipping mechanisms. So here what happens is your substrates come into the membrane, and they are even from cytoplasm, they come into the inner leaflet and they just get flipped to the outer leaflet. And that's how then you remove to the medium. And one of the ways this doesn't come back is because there is always sink outside, or either plasma proteins or others in the buffer, to which the drugs bind. And that's one of the reasons how these don't come back into the membrane right away. So there is always a gradient, and that's the flippage mechanism. Okay, so experimentally it's really difficult to really distinguish between the vacuum cleaner model and flippage model. So we assume both are really giving you the same effect. It's just a different way to explain the results. Okay, and then we also saw this on Monday, where I showed you that substrates come into the membrane from outside. They bind into the substrate binding pocket. We know that actually causes the ATP to be bound to the ATP binding sites. You get the dimerization of ATP sites, which actually leads to the major conformational change in your protein. And you go from here also, it's actually alternate access mechanism, because you're accessing the binding site from within the membrane. Once you open up, then it's sort of, instead of having inverted V, you have the V conformation. And experimentally, we have shown, others have shown that the binding affinities are much higher here for the substrate. But when you change the conformation, now the affinities change almost as many as 30-fold lower. And that makes sense, because when you want to release the substrate, you want to be able to dissociate faster. And that's why you have change in affinities when you have the ATP hydrolysis cycle continued. Okay, so this is exactly the same model, but now this is the latest one based on the double electron resonance that we saw yesterday with the more biophysical evidence, the deer. And again, you can see here the NBD is shown as, you have a range of distances, and then coming it together, the substrate binding, then you have the dimerization of the ATP sites. This is again, it's shown here with the two ATPs bound. We looked at the ATP sites, and I said that the ATP actually binds at the interface of the sites, right? And that's what is happening here. And then you hydrolyze the ATP sites, or the ATP to ADP, you release phosphate, you also release the substrate, and you continue the same alternate access model. So now with even biophysical techniques, you can sort of verify what we earlier knew from a couple of conformations. Okay. As most of the time it's not easy to do the transport studies. So we also look at then how we could use ATP hydrolysis. And here you can see you can use the enzyme kinetics now. You have the P-glycoprotein with ATP, which is the ES, E plus S. Then you get this conformation where you have the substrate or the ATP bound. You can also use a non-hydrolyzable ATP, which is the ATP gamma S. Once it binds, this is very tightly bound, and this is called non-exchangeable form or occluded nucleotide conformation. This is the step where actually the substrate changes its affinity. So even before the complete ATP hydrolysis occurs, your substrate is sort of moved from the binding site. And then this is just showing that the vanadate actually traps in this step. So the rate limiting step in this whole cycle is the release of ADP. If the ADP doesn't get released, then PGP doesn't go through this cycle. So most of the substrates you will see have effect on the ADP release step. Those which stimulate ATP hydrolysis release the ADP faster. That's how then you increase the velocity of your reaction. And those compounds which inhibit ATP hydrolysis release the ADP much slower, and that's how they affect the release. And so continuing yesterday's discussion, what we should be looking for is the modulators which would keep the ADP bound tightly or inhibiting the ATP hydrolysis. And if that happens, I think then we will have a better way to get a better modulator. And this we also looked at yesterday about the CryoEM studies, which again shows the same, that you have the conformational changes when you don't have any ATP. Molecules are going open and close, but then once you have the ATP hydrolyzed, you continue to do that, but then when you trap ADP, you have this closed dimerized molecule, release of ADP, and then movement in the NBD cycles, and then you go through this. So essentially we try to confirm all these things by using different techniques. You use your biochemical techniques, you use structural techniques, biophysical techniques, and then look for the evidence for supporting the conformational changes. Okay. So the next most studied example is this ABCG2 or BCRP, which is the HAP transporter, and the functional unit is the dimer or oligomer. Similar to P-glycoprotein, it's also expressed in canalicular membrane in liver, epithelial cell, apical membrane, small intestine, colon, blood-brain barrier, placenta. So when you look at the oral delivery of the drugs, you'll find that if you inhibit ABCG2, it has the effect on oral absorption of substrates like topotecan, mitoxantrone, anthracycline. And there are specific inhibitors for ABCG2, which is KO143 or Pimotrimorgan C. This is a fungal compound isolated from fungus. Okay? And then these are just some of the substrate we already looked at yesterday about the overlapping substrate specificity. Mitoxantrone or topotecan anthracyclines are also transported by P-glycoprotein, but antifolate methotrexate is not transported by PGP. Then fluopyridol will come to kinase inhibitors on Friday. And some of the HIV protease inhibitors also are substrates for this pump, and statins. So that's the drug-drug interaction with statin will have the ABCG2 will have a role in that interaction. So now we have two models for ABCG2 based on extensive mutagenesis studies which were just reported a couple of months ago, and this is the CryoEM structure of the transporter. Okay, so we have monoclonal antibodies people have developed for P-glycoprotein and ABCG2. So when monoclonal antibodies also can actually be used as inhibitory or modulators. You must have heard the term immune therapy because you have receptors on cell surface. You can get a antigenic Fab or antibody, you bind and you can inhibit. So some of these antibody as drugs are in use for treatment of breast cancer and others where there is you must have heard HER2 receptor. And Genentech actually first developed the antibody, but it's very expensive dosage. It costs almost, one treatment is almost like $100,000. So it's not something which is commonly used. So in this ABCG2, what happens is we know that the functional unit is a dimer. It's shown by two different colors here, blue and pink. And you will see that there are two Fab molecules bound. Okay, so when the two Fab molecules bind to extracellular region, the NBDs are kept close, and it doesn't allow it to hydrolyze ATP. And that's the mechanism of how the Fab inhibits the ABCG2 function. But you can bind the substrate. So there are two actually substrate binding pockets. One is this slightly bigger. There is a plug, they call it leucine plug, and then the extracellular. In the outer leaflet, there is another binding pocket. So this is without the Fab bound. You can see these are the loops. Substrate binds into the one first site. The ADP is hydrolyzed ATP. It moves on to the second site, and then it's transported. And maybe then, it's possible to use these Fabs as the modulators. But but somewhat different model came out of this extensive, more than 60 to 70 mutations were studied in different regions of this transporter, and they proposed this model where you have these NBDs, and there is a balloon-like flexible substrate binding pocket. And these actually, they think that then somehow this balloon, it squeezes out, and you release the substrate. But we don't really have direct evidence for either this model or this yet. But overall architecture seems to be similar. That is, at least the functional unit is homodimer. And this is the way it can function. As you can see, these are very recent studies, so we are still figuring out how exactly what happens. Then another class of transporters which are important are the MRPs or MRP1. One difference between P-glycoprotein and ABCG2 and MRPs is MRPs mostly actually conjugate the drugs with glutathione. So glutathione conjugated drugs are pumped by MRPs. MRP1, which is the ubiquitously expressed, actually also can transport leukotriene C. This is a cytokine. And interestingly, till now we did not really know what the binding sites were, how different were for leukotriene C and anti-cancer drugs. But recent structure, again this year, we have the structure of MRP, the bovine MRP1, and when they complexed with LTC4, because this transporter has a very high affinity for leukotriene C, it's like 1 to 2 nanomolar, so it was easier for them to get the complex structure with LTC4. And as you can see here, this binding pocket is bipartite. It has 2 regions. One is the polar region, they call it P pocket, and the other one is hydrophobic region, which is the H pocket. But most of the anti-cancer drugs actually only will bind to this H pocket because they do not have the polar region. So we don't know yet what would bind when you have anti-cancer drugs, but at least for leukotriene C4, you have this polar regions here binding the one half the molecule, and the second one is on the hydrophobic. And that's the N-terminal extension, which is totally independent from the basic transporter. Okay, and then as we were talking about the docking studies, this is how then you'll look at what the interactions are. Depending on the distance between these groups, you can infer whether there is a hydrogen bond interactions or van der Waals or what happens. Because to have 100 hydrogen bond interactions, this distance should be less than 3.5 angstrom. So if this distance is more than 5 angstrom, there is no way you're going to have hydrogen bond interactions. So this is mostly calculated based on distance. And the polarity of the residues are the groups which you have here. Okay? And as you can see, this is an arginine. So it is able to have the hydrogen bond interactions. So that's the overall structure of the MRP1 cartoon. But interestingly, what they found is that, as you can see here, we were discussing this yesterday, the NBDs are very far apart. But for some reason, when you bind the LTC4, now the NBDs are brought together significantly. And always this question we always ask, what's the signaling mechanism? How the substrate which bind, actually if you measure the distance between this and the ATP binding sites, it's 60 to 70 angstrom. So how does this signal get transduced? And we still are working on that. We don't have a clue for that. Okay, but this clearly happens. You can show by a variety of different methods. Basically, there are differences in the transport mechanism, the way MRP1 transports and P-glycoprotein, at least based on current knowledge. As you can see here, the drug, at least LTC4, actually is not able to partition from the membrane, but it comes from the cytoplasm, because these structures show that there is no really entry point from the membrane. It has to enter from the cytoplasm and then gets pumped. But in P-glycoprotein, there are some of these clefts, what they call portals, openings within the membrane, which allow the drugs to get into this open cavity. As you can see the open cavity here, but you don't really see here. There's no way to have, see that. And so that's why the P-glycoprotein pumps out from the membrane. So that's a clear distinction between how transport occurs. So this is about the mechanism of in vitro when you study the given transporter. So do you have any questions on this? Otherwise, I thought what I will do is cover some aspects of this under in vivo conditions and see what we learn or what we have. So as most of the drugs are given orally, you have the barriers to the oral drug delivery, right? From stomach, if you have the efflux transporters in your stomach or intestine, your oral bioavailability is decreased because of the efflux and then elimination in feces. And this also actually depends on the solubility of the drug, stability, and hydrophobicity. Then in your enterocytes, you have the absorption and then the drug metabolism, and then it goes to the liver where first pass metabolism, and whatever is excreted goes into your biliary efflux. Okay? And this is the interplay between intestinal epithelial transporters. Most of the secondary transporters are here. Then your basolateral is the MRP2, apical, MDR1, BCRP, peptide transporter, and others. And then these orally given compounds will be absorbed into the blood, or you have the efflux which will go into your renal portal vein, and that's just showing in the how it schematically occurs. So the pharmacokinetics consequences of induction inhibition of intestinal transporters. So some of the drugs do induce transporters, like rifampicin will induce PGP expression. So you need to be aware that if you give rifampicin, you are going to get induction of P-glycoprotein. And the inhibition of intestinal uptake transporters or induction of efflux transporters may decrease the absorption of drugs. And inhibition of the efflux transporters may increase bioavailability, and that's one of the functions that would be way you could increase the absorption or availability by inhibiting these pumps. But still we are far from selectively inhibiting some of these transporters. So P-glycoprotein is part of the biological barrier, like limiting drug entry across membrane in the cells of the target tissues. And as we saw, the target tissues are intestine, liver, kidney, blood-brain barrier. Those are the four. And the one of them, this is what you see is the dramatically seen in blood-brain, where we have the blood-brain barrier. We also discussed yesterday, I think, or day before yesterday, I told you about when they were developing the mouse colony lacking P-glycoprotein, they almost lost the whole colony because the mice which lack the PGP genes are hypersensitive, and this is what is experimentally shown. Then these are the normal mice. As you can see, the sensitivity, they are quite resistant to ivermectin, almost 100 milligram per. The IC50 would be something like 80, 90 milligram per kg, but those mice which lack the MDR1 and MDR, at least even a single gene, as we know in mice you have two genes, but even a single gene knockout actually becomes, the sensitivity is more than 100-fold. Okay. So you can also do in vivo, you would have heard what using F18 glucose for imaging. There are also other PET-induced imaging probes like System-AB is another one which is given to people, and then you can look at the tumor profile. Here this is just a mouse whole body autoradiograph for amprenavir, and the bottom line what you see is that amprenavir is actually, it does not enter the brain or the spine region completely. It's totally excluded from the brain, even though it's present in the in the intestine and other things. And this was one of the puzzling thing, because this is very good P-glycoprotein substrate from the Transwell assay. We looked at the polarized epithelial cells. So when that ratio is very high, 27, that means it's highly transported from basolateral to apical surface. Okay, but it's totally excluded from the brain. And then to show, how would you show this if it is really transporter mediated efflux from brain? One way is to show is that you use an inhibitor of your transporter. And so if you do now the scan again, this is the control that we saw earlier, and now you use an inhibitor like Electridar, or you can even use like a cyclosporin A, and now you can see you completely bypass the blood-brain barrier and you accumulate the amprenavir in brain. Similar studies you can also do now, the more recent models like zebrafish. It's very good, interestingly, the zebrafish brain has the same blood-brain barrier, and they have the similar transporters expressed. Zebrafish genes also, the PGP, has similar properties as human PGP and ABCG2. So now recently people are studying, instead of using mice, they can use zebrafish. because it's easier, and genetics is, is very well known, and it's easy to handle, and multiple experiments you can do. You can also see the such studies when being done with patients to get a quick assay if you want to know the tumor, whether it's localized to one organ or it metastasized. You can use F18 PET imaging. Unfortunately, I don't have an image of that, but there clearly it shows then if there are, let's say, if the tumor is going from adrenal gland, initial stages, and then it metastasized to lung, liver, brain, you can see then the deposits. We also looked at, I said you can also develop triple knockout mice, which is lacking two ABC, MDR1AB and ABCG2. We will look at the tyrosine kinase inhibitors as modulators on Friday, but since 2000, there has been this new approach called targeted therapies, and one of the first therapy which was started was development of imatinib or Gleevec by Novartis, and imatinib or Gleevec specifically targets tyrosine kinase and it inhibits its phosphorylation. And this was actually treatment was given to the chronic myeloid leukemia patients, or CML, and immediately they saw the much better effect and relapse. But as over the period of time, the patient started developing resistance, and we will see that most tyrosine kinase inhibitors get pumped out by P-glycoprotein and ABCG2, and that's how then you develop the resistance. But we'll come to that on Friday. But I just wanted to show you this in vivo data for these the tyrosine kinase inhibitors, and as you can see, this is all most recent one. Now only people have started using these tyrosine kinase inhibitors, and you have, this is just a schematic of blood-brain barrier, which we already saw. That number of transporters are present on the luminal surface of this surface of endothelial cells. And then these are some of the tyrosine kinase inhibitors like selumetinib, ponatinib. These are all actually are in the clinic used for treating patients. You will see some of the patients like having lung cancer, non-small cell lung cancer. They have some mutations in the tyrosine kinase inhibitors, and then they are treated with one of these inhibitors. And under those conditions, then if you look at those for these knockout mice, so this is the wild type, just 1A, 1B, that only two MDR genes are knocked out. This is ABCG2 alone. You can see there is slight incremental accumulation, but when you knock out all three genes, there is a drastic increase. So what it tells you is that when you are knocking out ABC, let's say MDR 1A or 1B, the function is compensated by ABCG2. The vice versa, when ABCG2 is knocked out, MDR 1A, 1B is able to take over that function. So only when you remove both these transporters, then you can see accumulation. And that's actually the first in vivo demonstration as far as the blood-brain barrier goes, how these transporters actually function together. And so that's the most difficult part, why having a specific inhibitor for a given transporter, why it's not going to work, because there are multiple systems overlapping, and we don't know yet, but this MRP4 or ABCC4 has very similar drug specificity as ABCG2, and so maybe next 5 years we might find that you also need to worry about MRP4 when you knock out all these. There might be some substrates or modulators which may be pumped out by MRP4. I mean, in that case, then you'll have, because we already have the MRP4 knockout mice. It's not that we don't have this quadruple one where we knock out all of them, but that's just to show you the status. Okay, and why this is actually the in vivo demonstration why it's important to have study both these transporters, why FDA wants you to study these transporters. Okay. So should we move on to the next one? So this is one of the topic, as you will see once you have your drugs being developed, what the regulatory affairs people will be talking about or what are the requirements. So unfortunately, I couldn't transfer the PDF files of this one of the presentation, but I will email it to you, or Uma will give it to you later. So I think in 2007, they actually established the International Transporters Consortium, ITC. So as the transporter field is increasing and people found that, as you will see, you also saw throughout the presentations, the nomenclatures, so earlier you used to just people name the transporters as they wanted to, but now there is a uniform, like at least for ABC transporters, SLCs, uniform nomenclature. And those came through having a consortium of people working in the field and the consensus. So that's one of the reasons why this International Transporters Consortium was formed, mainly by pharmacologists and those working in the industry, because they wanted to have a common platform. So the results can be explained properly and you can plan experiments further for your new drug entities. So they had 2 workshops. I think this year there's another extra workshop was done, and then they recommended their recommendations to the FDA and also the European Medicines Agency. So this was actually the first sort of a draft guidance from FDA in 2012. A number of transporter-based interactions, they were documented in the recent years. So this is when actually they started putting on the label of the drugs that some of the interactions were found with transporters or whatever when they had the drug-drug interactions. All investigational drugs should be evaluated in vitro to determine whether they are potential substrates of P-glycoprotein or ABCG2. In older literature, or another alternate for ABCG2 is the BCRP, breast cancer resistance protein, or MXR, because the gene was cloned from mitoxantrone resistant cells, so they named as MXR1, but ABCG2 is the standard. Investigational drugs should be evaluated in vitro to determine whether they are substrates also of hepatic uptake transporters like organic anion transporting polypeptide 1B1 or 1B3 when their hepatic pathway is significant. And then similarly, investigational drugs should be evaluated in vitro to determine whether they are substrates of OAT1, OAT3, and also organic cation transporters. So most of this information actually is available at the FDA website. It's freely available. Anybody can access it. Okay. So when you look at drug-drug interactions, you'll find that there is we actually saw some of the overlapping substrate specificities for transporters. But as you know, the cytochrome P450s are involved in metabolism of drugs and toxic compounds. There also is a overlapping specificity for the CYP3A and P-glycoprotein. Similar thing you can find for ABCG2. And these are some of the drugs, interestingly, which are metabolism by the CYP3A and transport by PGP. And that's why you need to be aware of these common features. People have tried to actually identify the binding sites for these substrates on especially the CYP3A4 isoform, because it's very close to PGP. Unfortunately, those efforts have not really yielded any new information, because ideally you might think that if like paclitaxel is binding to CYP3A4 site, it may be similar to what you have in P-glycoprotein, but those studies haven't really been conducted to that extent. There seems to be a stability issue with the CYP3A4 enzyme when you purify it. It's also a membrane-bound enzyme, so that's another complication there. Okay. So if you go through this FDA website, you will see some of these decision trees. So all the the new chemical entities, are they interacting with PGP or ABCG2? Is the hepatic or biliary elimination more than 25%? If it is, then definitely it's transporter mediated. Is the renal elimination more than 25%? And then what you need to test. And then further guidance is that additional studies may be needed depending on knowledge of structurally similar drugs in the same therapeutic area. And then you have this guidance PDF. So what happens if you have your compound substrates for PGP or ABCG2, then you have this decision tree. You may want to do transport studies, and if the net flux is more than 2, which means it's being transported from basal to apical surface, you may have to go through this efflux, testing that. If the net flux is ratio below 2, which means it's not really interacting with PGP or ABCG2. And so, it just walks you through the whole system. It's mainly for those involved in, developing drugs in industry. And then some of these are the potential clinical inhibitors like quinidine, verapamil, etranacozole. And this is given, I think you have the PDF file of this. This was the first report actually when they started this. It's similar to the transporter substrates, you also have then the PGP inhibition decision tree. And I'm sorry, but it's not really that clear, but essentially you will look for how the function of the PGP or ABCG2 is inhibited or not, depending on your transwell studies. And then if you are, do the pharmacokinetic studies, then you need to look at the IC50s, EC50s and ratios. And then you also look at the mean steady state total free and bound maximum binding following administration of the highest proposed clinical dose of inhibitor. And this is more to determine what the dose you should have. So the drug discovery process is always a balancing act. You will go from chemistry, biology to biochemical screen. If that doesn't work, you always need to go back to your chemistry, biology to fine tune the structural motifs. Then you move on to the cell-based screen. If there are changes or differences, you go back again to the chemistry, biology and come back to, then the optimized conditions, then you go to the in vivo or non-clinical studies. Then you have the IND enabling studies, clinical trials. That's where then you could complete the mechanistic or non-clinical studies. It doesn't have to be done at the earlier stages, because if it doesn't come up to there, then it's not needed, because these libraries, when the companies screen, they are looking at 500,000 or 1 million compounds at a time. And so it's not easy to really go through whole process for entire screening process. And then you have the clinical trials and then regulatory approvals. But actually once phase 0 or phase 1 clinical trials start, the companies they are actually in contact with FDA, because they want to go back and forth and provide all the data so that FDA can tell them what needs to be done, how it should be done, and they walk them through the whole process. So that's more now recently becoming more efficient and cost effective, because if the FDA has a concern at phase 1 clinical trial, the company wants to actually resolve then and there that stage, rather than going to phase 3 trial and coming back, the FDA saying, oh, we are not going to approve your drug. And so this is more becoming a partnership between the companies and FDA. And I'm sure the same thing happens in European the medicines agencies, because they want to work together. Okay. So the summary of this, the section is the drug transporters can have a major role in determining the pharmacokinetics of drugs. So this was in 2012. ITC FDA has identified 7 transporters of key interest during drug development, and we'll see, just last month they added 2 more. Drug-drug interactions mediated through drug transporters have received increased attention because of the clinical reports of toxicity and other adverse effects. And then evaluation of in vitro screens to predict in vivo drug-drug interactions is an area of increased awareness. So the FDA actually doesn't have any problem if you provide data with in vitro studies, so long as further down the road there is no effect when in vivo studies are conducted. Okay. So this is actually the PDF, the presentation by Li Zhang. She is an FDA senior advisory for regulatory programs and policy, Office of Clinical Pharmacology. So they have a website, and you can actually go through this entire presentation. It's very nicely presented, and she actually gave this presentation at the, in Washington DC on March 14 this year. This is again to make industry people and those working in the field aware of what's the current thinking of FDA has about the transporter mediated drug-drug interactions. And so in nutshell, I'll present some of the things, and then you can look at the PDF of this. Drug transporters, as we discussed so far, contribute to variability in drug concentration and response, and the pharmacokinetic determinants are the absorption, distribution, metabolism, and excretion. This is the ADME. We keep coming back to this theme. And then the pharmacodynamic determinants, delivery to the site of action, control of tissue concentrations, therapeutic targets. So we discussed this a little bit. The sodium glucose co-transporter inhibitor for the treatment of type 2 diabetes. And then the urate transporter inhibitor in combination with an exo inhibitor for treatment of gout, because this is now, as we discussed, is transported by also the ABCG2, and we have the polymorphic changes in ABCG2. Okay. Which transporters are clinically important and should be considered for evaluation during drug development? So, those which affect drug-drug interactions, then also the genetic variation of the populations, and we'll see some of this tomorrow's when we look at the polymorphic changes in transporters, then the disease state, and finally the safety and efficacy. So these are the main factors that FDA looks at. This is just a chronology of how they started developing this. So FDA first published the draft drug interactions guidance in 2006, and then you can see now actually FDA has an independent clinical pharmacology advisory committee, which earlier they did not have. And now this 2007 we just discussed the ITC formation, and then the first the expanded guidance came in 2012. Third workshop they had just this year, and this is the October of this year they had this guidance, and I think next year they will have the expanded version. So what are the emerging areas as far as FDA is concerned in this field? So altered drug concentrations in tissues, knowledge of drug as transporter, substrate, or inhibitor may help to explain some observed clinical effect. For example, statins and metformin in liver tenofovir, cisplatin in kidney. We discussed about toxicity to kidney as well as the metformin and statins being substrates for some of the transporters. Then you have the altered endogenous substrate concentrations where increased serum creatine by a drug may not represent renal toxicity. So these are some of more clinical questions. Bile salts and conjugated bilirubin are transported by. So BSAP is the ABCB11. So this is the one of the only transporters which is highly specifically expressed in liver. Most of the other transporters are in different tissues, but BSAP or B11 is only expressed in liver. Non-clinical and clinical experiences during drug development, which will dictate whether to evaluate these transporters. And then the coporphyrins, CP1, CP3, or in addition to these being transported by these anion transporters, they also interact with ABCG2. That's recent studies have shown, and so they need to be looked at. So alterations in transporter activities contribute to variability in pharmacokinetics and pharmacodynamics, efficacy and safety. In vitro transporter studies increase our ability to predict occurrence of in vivo drug-drug interactions. And that's why FDA allows you to do the in vitro studies. And then the decision criteria proposed are being used to predict DDI, drug-drug interactions. And then now recent development is this in silico methods, PB, PK have potential utility. So they are actually encouraging, the FDA would like you to do more of the in silico analysis because it's time saving and it also may give you more, the principles which can be further down tested. And as with any other field, the transporter field research is also evolving as we learn more new things. So the emerging transporters with clinical importance may need to be considered as we develop, describe or learn more about properties of different transporters. This will need to be changed, and then the transporter's sole like a role in toxicity or efficacy needs to be understood. For example, the organic cation transporter one. And then the question is always whether can we use transporters as biomarkers? Is that possible? And some of the studies are going on. So you may have heard term gene signature. So there are now you will see more than 50 to 100 papers where what people do is they take say 100 or 200 samples of ovarian cancer or breast cancer, isolate messenger RNA, and they have these chips with genes depending on what you're looking like. We have a chip developed for genes linked to multi-drug resistance. So there are 300 genes, 380 genes which cover all the cytochrome P450s, all the ABC transporters, all the SLCs, and other enzymes which metabolize drugs. So in one single chip, and these are all 384 samples you can load at one time, you will get to know in your patient population what the expression level of your transporters are. And then you always compare with the control population. And so if you have more than 100 samples, these can be statistically significant. And then you look at the pattern, so your cutoff is, so expression level is if it is more than, let's say, twofold or threefold, then you take it as a significant difference. And by that, when people look at it, you'll find that out of 380, maybe there may be 5 or 10 or 15 genes which show increased expression or decreased expression. And so these are then called, what is called then the gene signature for a given type of cancer. And then the next phase people try to do is out of those 10 genes, you'll say, okay, 3 are overexpressed mostly. So then you validate that and see whether that will give you a result which is similar to the what you see in tumors, which may not or may correlate depending on interactions. Okay? So we are still far from really coming to this state like where we can identify or use transporters as biomarkers, because although we know the ABC transporters are expressed in breast cancer. But when you do the gene signature from patient samples, you don't really see any of the ABC transporters coming in top 10 list. So it's not, the easy, but you also don't know what the patient population is and everything. So that needs to be.
When we screen, actually, how much is the consistency? So, for example, if we screen 10 patients,
Yeah.
100 patients, so the ABC transporters are overexpressing almost 100?
No, they don't look at this is just the patients who come in the clinic, and they show you the disease conditions, and then they have the samples. So these are more retrospective studies. It's not like you designed the study for this. This happened to be centers treating for those, treating those cancer patients. But they store these biobanks, right? They're having the tissue. So then people go back and say, okay, we have these samples. Can we look at these profiles? So though the prospective studies have not been, done yet, where you ask these questions, then recruit patients, it's almost going to be impossible to find that many patients in a given city or. So this is the most latest guidance from the FDA. So this is just so you can see the comparison. 2017 draft, which is last month, and this is the 2012 draft. Okay, so major drug-drug interactions transporters. So you have the PGP, ABC, G2, then some of these are organic anion transporters, organic cation transporter, and these are the two which they added new, MATE1 and MATE2, okay, which were missing from this. So that's how we have come to now 9 transporters. Earlier we had 7. And then study design for transporter, substrate, and inhibitor assessments. So they say now that you should have more detailed and transporter-specific recommendations on study design, test system, data analysis, and interpretation. See, earlier it was only briefly outlined in decision trees. Now they are given more specific guidelines. In vitro induction study, actually now earlier they wanted you to consult with FDA, but now they don't want you to really look at that. Timing of studies dependent on therapeutic indication. Instead of, not discussed, now they say it depends on what the effects you may find. Considerations of metabolites, drug-drug interaction potentials toward transporter should be assessed on a case-by-case basis only. It's not that every entity has to be tested. And this was earlier criteria, which is that major circulating should be more than 25% of the parent. Test system characterization and validation, general recommendations on qualifying test systems are not discussed. So mostly in vitro systems are now well accepted, and FDA will allow you to use those. So this is how, then you can go ahead and do what, if you end up working in the industry sometime, you'll have to go through these things. And so most pharmaceutical as well as biotech companies now, when they are trying to develop drugs, in their preclinical drug departments, they have instituted the drug transporter studies. They always screen compounds for transporters. Earlier this was not the norm, but now it is because of the FDA regulations. And it's also cost effective, because 10-15 years ago, when people were doing clinical 3 trials, they came to know that the entities were substrates for transporters, and then they had deaths during clinical trials and high toxicity. So that's really very costly decision, because you waited till clinical, 3rd clinical trial, phase 3. And so now, most companies are aware that transporters are important. So, you have any questions about this? Because, more information about this at this stage, I'm not sure it's really relevant, because unless you are going to be doing those things, it's not going to be really that useful. But the other reason not to go into details is these are all readily available on the website of FDA, and they are not the only, like they are, what is, these recommendations are only as of today. They would change, in next 6 months, 1 year or whatever. So these things are all changing. So you have to just, as they go along. But this was just to give it to an idea about you, how the process works, what is involved in drug discovery, and how, why it takes so much time, why it takes so much, cost-wise, and all the efforts. Because the prime factor which is considered is the safety and also the non-toxicity, because that's most important, or relatively non-toxicity, because there are always some side effects, although we may not come to know when the drug is introduced, but over the period. Okay?